You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is December 2nd, 2024. Our topic is Abbas Chapter 7, Immune Receptors and Signal Transduction. This is presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine, Pediatrics, and is a Chief in the Section of Allergy Immunology at Children's Mercy Hospital in Kansas City, Missouri.
Today we'll be talking about Chapter 7 from Abbas. We'll be talking about immune receptors and signal transduction. Today is December 2nd, 2024. I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City. And my email is included if you have questions or comments. All right. Some disclosures, nothing pertaining to the talk today, and the learning objectives are to understand the signaling for T cell receptors, B cell receptors, and cytokines. Pre-test questions here. Which of the following is an example of GPCR? In T cells, calcium signaling pathway induced by antigen recognition, which enzyme and transcription factor are activated? All right, so this chapter you'll be learning lots of new concepts and terms. So I want you to kind of keep your minds open to learning kind of a new concept. Really, I want to kind of go over the basics of signaling, and then we'll dive into T cells, and then we'll compare that to the B cell signaling and talk about cytokines. So, as I have mentioned in the past, for any signaling pathways that we learn, I want you to understand the initial terminology and why there is signaling that's required, and then knowing the end result, because the end result connects to the initial receptor binding to its ligand and why that happened. So kind of keeping that end goal in mind will help you understand the overall signaling. So what do we mean by signaling? So the receptor and ligand interaction starts typically on the surface of the cell, where the ligand binds to the outer part of the receptor, and then there is that cytosolic phase where there are interactions between different molecules, and then later there is a molecule that moves from the cytoplasm to the nucleus, and that's called the nuclear phase. So in the signaling, there is a receptor ligand interaction that takes place, then there is a cytosolic phase, and then there is that nuclear phase. In some interactions, there is no transcription. In some interactions, there is no the initial receptor binds to its ligand and the nuclear phase starts directly. So there are different types of variations, but for our discussion, we'll stick to this pattern of receptor and ligand interaction, then the cytosolic phase, and then the nuclear phase. If there is no transcription, we are talking about maybe other kinds of changes like cell motility, granules, exocytosis, apoptosis, or protection from cell death. So lots of different end results that can occur after signaling. So the first phase is the receptor and ligand interaction. So as I mentioned, most of these that we talk about are cell surface receptors, but there are also some nuclear receptors. So these have to be lipid soluble for them to be able to be working on the nuclear side. And then when that receptor binds to its ligand, there is a novel geometric shape that's created. Either there is a conformational change in that receptor, or there is cross-linking, so lots of receptors coming close to each other that gives rise to other processes. All right, so talking about receptors, there are different types of receptors. There are certain type of receptors where there is modification of the receptor or non-receptor molecules in the close proximity. When there are several different types of processes that can occur for modification of the receptor, such as phosphorylation, dephosphorylation, ubiquination, acetylation, or methylation. Again, for immune receptor discussion, our big ones are phosphorylation and dephosphorylation. And so the phosphorylation, when there is an enzyme that is involved in phosphorylating the receptor, these enzymes are called kinases. So kinase is an enzyme that adds a phosphate molecule. When the phosphate molecule is added to amino acids of the receptor, they are called protein kinases. If they are added to lipid molecule, they are called lipid kinases. So there are different kinds of kinases. Kinases means that they are adding a phosphate molecule. Now, because the phosphate is in protein kinases is added to one of the amino acids, if they are added to a certain type of amino acids, they are named that way. For example, if it's added to the tyrosine, it's called tyrosine kinase. If it's added to serine or threonine, they are called serine or threonine kinases. So you get the point. Kinases means that there is a phosphate molecule added. Protein kinases, lipid kinases are different types of kinases. If dephosphorylation occurs, which means if there is a phosphate that's removed, then the enzymes that are needed for that kind of modification are called phosphatases. And then so on and so forth. So there are several different types of modification. So if the phosphate molecule or the tyrosine is part of the receptor, it's called tyrosine kinase receptor. But there are some receptors where the tyrosine is not on the tyrosine kinase is not on the receptor itself, but associated with it. So when that happens, that receptor is called non-receptor tyrosine kinase based receptor. Right? So it's not on the receptor, but it's a receptor that still takes advantage of this tyrosine kinase in the close proximity. So those are two different types of receptors. Then you hear about these other receptors called nuclear receptor, which means it's actually not on the surface. Nuclear hormone has to pass the lipid membrane, so the cell membrane, and get into the cytoplasm where the nuclear receptor is present. The hormone binds to the nuclear hormone receptor, and then it crosses the nuclear membrane where it will help in transcription. So that's your nuclear hormone. GPCR is a 7-transmembrane receptor where activated G protein activates adenyl cyclase and phospholipase C. And these will help in ATP converting to cyclic AMP, and then the G protein itself is taking part in that process. So this is another type of receptor, and then there are receptors that lead to proteolytic cleavage of receptor, which means it pretty much breaks the receptor. So here is the pink receptor that it has the external or extracellular part of it, and then the intracellular part of it, and those are connected by a small kind of a structure. And when the ligand binds to that receptor, there is cleavage of that extracellular portion, and then the intracellular portion detaches and can move to the nucleus. So different types of receptors have different kinds of roles. We talked about kinases. When you think about this terminology, there are a lot of new terms here, and I want you to kind of think of say modular furniture or Lego pieces, and just like any kind of modular furniture, you can put different pieces together to form different shapes, or you can put Lego pieces together to form different structures. The same way, there are structures or domains in these immunological molecules that come together to form various enzymes or different types of adapter molecules. So we talked about these non-receptor tyrosine kinase receptors, right? Which means that the tyrosine kinase is on a separate structure in close proximity to the receptor. So there are different types of these tyrosine kinases, and they are classified into families called SERC family, SIG family, TEC family. So all these names come from the kind of the domains that are involved in it. SERC contains various, here is the SERC family kinase right on the top here, SIG family kinase, and you see the structure is different where these SH2 domains, SH3 domains come together to form the structure. The kinase part is common to all three of them. But the rest of the structure is different, and that determines what kind of receptor or what kind of structure are these going to bind to. So SERC family kinase has one SH2 domain, think of Lego piece or like a part of the modular furniture that's binding to the kinase part. Then there is this SH3 domain, and each of them have are attracted to a particular type of molecule, such as SH2 domain can bind to phosphotyrosine, SH3 domain can bind to proline-rich peptides, and then pH domain is another one that can bind inositol phospholipid. So these structures are our Lego pieces. And when the kinase is bound to these various different types of Lego pieces, it determines what kind of receptor it can phosphorylate. This kinase will phosphorylate a structure, but that structure has to be bound to the rest of this tyrosine kinase. So if there is a peptide that has phosphotyrosine and a proline-rich peptide, it can bind to a SERC family kinase that can bind has both SH2 and SH3 domains. SIG family kinase are the ones that have two SH2 domains, which mean they might have tandem phosphotyrosines that can bind to them. And then the TEC family kinase has that pH domain that binds to inositol phospholipid. It also has SH2 domain and SH3 domain, so it will determine which kind of structures it will bind to. Right, so these are different domains that come together and determine what kind of structures they will bind. And because these domains are part of these tyrosine kinases, they can be classified into families called SERC family, SIG family, and TEC family of kinases. Hang on to that thought, and then we'll come back to these kinases in a little bit. Now we're going to talk about more structures that are called adapter proteins. And just like we use adapters, right? When we use adapters for our electronics, we are just trying to connect two things together, right? They don't really give the output or the input, but they are just connecting two electronics together for them to be bound to each other and function together. Same thing with these adapter proteins. These adapter proteins are just docking sites. They physically link different enzymes or different molecules so that they can bring them together and they can function together. These could be on the surface in a membrane protein or in the cytoplasm. So couple of again, these adapter proteins have various domains such as SH2, SH3, proline-rich regions, tyrosine residues as a part of their structure that determines what they can connect. And so examples of these are LAT or SLP76. These are just adapter proteins that are bringing other structures or other enzymes together so that they can function. All right, so we talked about signaling proteins like kinases. We talked about adapter proteins. Now, before we go on and talk about more with that background, let's talk about immune receptor families. So again, there are lots of different immune receptors, different types of immune receptors, and such as PCR, TCR, FC receptors, PD receptors, and a lot of them, as you see, have this structure that we are very well kind of introduced to in the past, called immunoglobulin superfamily region, right? So this globular part is a repeating structure in all these receptors. This kind of region will help in ligand binding. It's external to the cell where it's going to bind to the ligand. Then there is that transmembrane signaling protein that helps in taking the message from receptor ligand binding to the interior of the cell and then eventually the nucleus. And so one such protein we know is the tyrosine. So a lot of these signaling molecules, such as for B cell receptor, these signaling molecules Ig alpha and Ig beta have tyrosine in the cytoplasmic tail of it. TCRs have signaling molecules or signaling chains like epsilon, gamma, delta, zeta, all these chains, or CD3 and zeta chains that have tyrosine containing motifs in it. Similarly, for Fc receptors, Fc gamma R2b, PD1, and as you see, the tyrosine containing motifs are a little bit different in the ones that we talked about with TCR PCR versus Fc gamma R2b and PD1. And the big difference is these two receptors are inhibitory in nature versus the T cell, B cell, Fc, some of the Fc receptors are activating receptors. So these motifs in the cytoplasmic tail are known as ITAMs or ITIMs. ITAMs means immunotyrosine receptor motif with that is activating, or I stands for inhibitory motif. As you see, the structure of each one is different, where the ITAMs have tyrosine, several other amino acids, and then leucine, isoleucine that are included, versus ITIMs have valine, leucine, and isoleucine. So that valine is part of that, which is very different from the ITAM that is inhibitory in nature. So both of these motifs have important roles in immune receptors. All right, so that's ITAMs and ITIMs. Give me 1 second here. All right, so again, something to remember, and then we'll talk about these ITAMs, which is immunoreceptor tyrosine based activation motif and inhibitory motif, and we'll come back to these. The importance of these is that these are close to the Circ family kinases. What do Circ family kinases do? They help in phosphorylating the tyrosines. So when these receptors, all these immune receptors that have tyrosine motifs in their cytoplasmic tail are in close proximity to Circ family kinases, those kinases, the enzyme, will help in phosphorylating these tyrosines in these motifs. All right, let's talk about the overall signaling for lymphocytes. So thinking about the signaling, if you think about a switch, there are a couple of types of switch. It's an on-off switch, which will turn the signaling on or off. But then there are also the newer switches that have graded response, and not just on and off, but a graded kind of increasing, increase in the signal of that of the strength in these receptors. So thinking about the signaling during the maturation of these T cells and B cells, there is a difference in how they react. And because, as you saw in the previous slides, there are several signaling molecules with multiple ITAMs or ITIMs that have several tyrosine motifs. These ITAMs can be used progressively in T and B T cell receptors, so that there is a kind of increasing or decreasing signal strength with more use of number of ITAMs that are used or ITIMs that are used. Similarly, along with the receptors, the co-receptors CD4, CD8, or in case of B cells, CR2 or CD21, these co-receptors also have items in them. Inhibitory receptors for T cells are CTLA4 and PD1, or for B cells Fc gamma R2b, and we saw that they have items in them. And then there are some co-stimulators, or not just the co-receptors, there are co-stimulators that provide that second signal, and that second signal such as CD28 binding to CD8086 also has that kind of use of items that can help in determining the strength of the receptor ligand binding and leading to the signaling. Thinking about that, about the TCR, I know we talked about it a little bit. This is just kind of more going into the depth. We know that the TCR has alpha and beta chains, and then there are epsilon and gamma chains, or epsilon and delta chain, that come together to form CD3 molecules. Along with zeta chains, they have multiple items in their cytoplasmic tail. This table here shows the differences between TCR and then the BCR, which is the immunoglobulin molecule, and both of them have their signaling molecules CD3 and zeta chains, and in case of B cells, Ig alpha and Ig beta. And just kind of pointing that out. Here is the TCR complex in a different view. You see that the immunoglobulin super family structure there, and these TCR complex, if you look at that view, it has that beta pleated sheet view, and this TCR is going to come in contact with the antigen presenting cell that's presenting the antigen or the peptide bound to the MHC class 1 or class 2 in that case. And so here it's just showing class 1 with beta 2 microglobulin and the alpha chain interacting with the T cell receptor along with that peptide. Talking about the co-receptors, once that happens, it's an important thing to remember is that the CD4 and CD8 receptors. So if you look at this cell membrane, think of this is the APC, antigen presenting cell, that has the MHC class 2 on its surface, and then it has that peptide that it's presenting. This is the T cell on the top here in the panel B that has the TCR alpha beta interacting with that class 2 MHC. This is class 2 MHC interacting with a T cell, which is a CD4. So CD4 is the co-receptor that's also going to interact with that MHC class 2. And then on the cytoplasmic tail of that is a Circ family kinase. So co-receptor is in close proximity with the T cell receptor in CD4 cell. The CD4 in its cytoplasmic tail has the Circ family kinase called LCK. What does LCK do? LCK is a kinase, which means it's going to phosphorylate tyrosines. Tyrosines are in the cytoplasmic tails of the signaling molecules attached to the TCR. So LCK is going to phosphorylate some of these items here. That's where the process of signaling starts, when that interaction between the antigen presenting cell and the T cell receptor starts. All right. So one more this is the last slide before we start talking about the actual signaling in T cells. So I'll stop after this slide here, but just want to show what happens on the surface of the cells. When the T cell comes in contact with an APC, APC presents the peptide attached to class 2 MHC to the TCR. When that happens, the CD3 molecules and zeta chains are in close proximity to the TCR, that is in close proximity to the CD4 that has the LCK on its cytoplasmic tail. CD4 also interacts with the class 2 MHC. When this interaction is happening, these two cells have to be in close proximity to each other and bind to each other, adhere to each other. And that happens because of the adhesion molecules, which are our integrins and selectins, right? So these integrins, LFA1 is interacting with ICAM or integrin ligand, so that these cells or the interaction between these two cells is stable. There are several. We talked about the T cell receptor, the co-receptor, and then the co-stimulator is also playing a role here, whereas interacting with the CD28 is interacting with P71 and B72 on the APC. The other inhibitory molecules that can take part in that are CTLA4 and PD1 that are interacting with the ligands for those particular surface molecules as well. So all of these interactions are occurring between two cells, APC and T cells, and so this whole process occurring at this junction of these two cells make a cluster on the surface that's called supramolecular activation cluster, or SMAC. There is the central part of the SMAC, which is the interaction between the main interaction that we are interested in, which is the TCR and the co-receptor and the signaling molecules, the co-stimulators. But then there is also the peripheral part of that that's keeping those two cells bound to each other, which is the peripheral SMAC. So supramolecular activation cluster is just that interaction between two cells that is occurring at the surface of these cells. Now, keeping in mind that there are these lipid rafts or glycolipid-rich domains that are pretty flexible and can move and make this whole interaction pretty flexible. So the SMAC has important functions. Yes, it keeps the contact between the T cell and the antigen presenting cell pretty stable. But in case of CD8 cells, it might be helping in delivery of granules between the cells. In case of CD4 cells, it might be helping in the delivery of the cytokines between these cells. The signaling molecules are delivered to lysozymes and endozymes for degradation, and that helps in terminating the T cell activation. All right, so with that in mind, I'm going to stop here to see if you guys have any questions for me in relation to all the terms that we talked about. Right. If not, we can go ahead and move on. With that basic information, we'll move on to talk about the signaling in T cells. So we're going to go back to the same concepts that we already talked about and see how those things play a role in T cell signaling. So the first phase of signaling is receptor and ligand binding. So that leads to the end result that the T cell signaling occurs so that there is T cell clonal expansion, differentiation of that cell, that's activated T cell, to say a CD4 helper subset, Th1, Th2, Th17, or a CD8 T cell to become a cytotoxic cell, CTL, right? So the differentiation occurs, and then the effector functions take place, whether there is release of cytokines or cytotoxicity. During this time, it's important that the signaling also helps with prolonging the survival of that activated T cell so that it can provide these functions. So the end result of receptor binding, T cell receptor binding to a ligand, an antigen, is that the T cell can get activated and undergo clonal expansion, differentiation, give effector function, and prolong its own survival. So to do that, we need that overall signaling that needs to take place. And so what happens with that is when the receptor binds to its ligand, there is a novel geometric shape creation. There might be cross-linking of T cell receptors on the surface and then a conformational change. And when that happens, there are various signaling molecules and enzymes that interact with each other downstream of that receptor ligand binding. The end result of those signaling pathways is activation of transcription factors. Once the transcription factor is activated, it moves to the nucleus. It is translocated to the nucleus, and it binds to the promoter region of specific genes where it can affect the gene transcription. When that gene transcription ends making mRNA, mRNA will code for proteins that are needed for the end result. End result, remember, was this. The end result, say, requires IL-2 gene. So that this signaling will lead to transcription factors that will bind to the promoter region of IL-2 gene. End result is that mRNA is created that will code for IL-2. IL-2, when it is released, it will help in T-cell clonal expansion, differentiation, its effector function, and it help in survival of that activated T-cell. So the end result is what finally happens because of that receptor and ligand binding, and all of the signaling pathways are just a process to get to that end result. So some of the transcription factors that are important to know for T-cell receptor signaling are NFAT and NF-kappa B and AP1. AP1 has two parts of it, CFOS and CJUN. So if you think about it, we know that once the transcription factors are activated, those factors are gonna go to the nucleus and help in, IL-2 is just an example. It will help in gene transcription of several different proteins that are gonna take part in T-cell function. So our job is to figure out how does the binding of ligand to the receptor end in activation of these receptor transcription factors, and these are the three transcription factors that are important. So that's our end goal, how the signaling ends up in activation of these three transcription factors. So let's think about that process of TCR binding to its ligand, right? Here's an APC, here's the T cell. The MHC is presenting a peptide to the T cell receptor, and when that happens, the T cell receptors cluster together for cross-linking. Along with that, that cross-linking occurs because there are lipid rafts that move those receptors to closer together. Anyways, once that happens and the receptor is bound to its ligand, there are several signaling molecules that are in close proximity to the CD4 or CD8 co-receptor. The LCK on the cytoplasmic tail of the co-receptor is gonna phosphorylate the items on the cytoplasmic tails of the signaling molecules, CD3 and zeta chains. Once that phosphorylation occurs, there is another molecule. So say here is our zeta chain. It phosphorylates two tyrosines. Now ZAP70 is a molecule ZAP stands for zeta associated protein. So zeta associated protein 70 is the one that's an adapter protein that can bind to these phosphorylated tyrosines. When that happens, ZAP70 will interact with something called LAT. That is also a adapter protein, but LAT will act as a docking station for multiple enzymes, PLC gamma 1 pathway and MAP kinase pathway. So there is assembly of various enzymes and adapter proteins on these scaffolds, and there are signaling pathways that get activated. So again, this is early part of signaling, peptide MHC complex interacting with TCR, TCR interacting with the co-receptor interacting with that interaction of MHC and TCR. LCK on its cytoplasmic tail will phosphorylate the items, and those items on the zeta chain will interact with the ZAP70 protein. ZAP70 will phosphorylate, will interact with the phosphorylated LAT. So once that happens, there are various various pathways that will get activated, including PLC gamma 1 and MAP kinase pathways. So to remember these these sequential changes, you have to remember these molecules. And one of the simple, easy, and funny mnemonic that people use are a baby licked the floor, so got zapped and went lateral. Pretty inappropriate, sorry about that. But in general, it works pretty well to remember that mnemonic of lick, zap 70, and lat. Those are the interactions that occur. Remember, these are just the major pathways that we are talking about, but there are similar interactions occurring, such as instead of lick, fin is another enzyme that can phosphorylate tyrosines in items. So lick or fin, zap, interact with the phosphorylate the items, and then zap 70 interacts with those phosphorylated tyrosines, and then also binds to and phosphorylates the adapter proteins, including lat. Once that lat interacts with various proteins and enzymes, it is a docking station for several of those. There are pathways like PLC gamma 1 and MAP kinase that get activated. Why are these pathways important? Because MAP kinase pathway leads to activation of transcription factor AP1, and PLC gamma 1 pathway diverges into two pathways, DAG pathway and IP3 pathway. DAG is important for NF-kappa B1 transcription factor activation, and IP3 leads to NFAT transcription factor activation. So those are our three major transcription factors that are going to be activated through this initial process. So now we'll go into each of these pathways. Please stop me if you don't understand something, because it's easier to tell you, because you're going to continue to progressively add on to this information. So just if you don't understand anything, let me know. So again, after this initial signaling, these are the three pathways that we're going to talk about. And then the fourth pathway that I want to make sure you understand is about the survival of the T cells. So 3 transcription factor activation and survival of T cell is the end goal of this signaling. So the first one we'll talk about is the cell survival. So PI3 kinase is activated because of this initial receptor ligand activation, and then PI3 kinase will lead to phosphorylating PIP2 to PIP3. PIP3 is a structure that will lead to activation of a enzyme called PDK1. So PDK1 activated form will activate AKT. Activated AKT will inactivate apoptotic factors such as BCL2. Because it's not going to undergo apoptosis, this cell will survive longer. So PI3 kinase pathway is an important pathway because deficiency of PI3 kinase, as we know, can lead to immunodeficiencies. So this is an important pathway to understand. All right. So again, going back to our initial signaling, we'll just revise that. TCR binding to its ligand. Co-receptor CD4, for example, has LCK that will phosphorylate the items. Zeta chain, once phosphorylated, will interact with ZAP70 that will phosphorylate LAT. LAT will lead to the bringing in various molecules together on its as it acts as a docking station. And so two important ones are molecules that come together are called GRAB2 and SOS. GRAB2 SOS interacts with RAS to make RAS GTP. RAS GTP is going to help in interacting with a couple of other molecules, RAF, MEK1, and then ERK1, ERK2. Activated ERK is going to go to the nucleus and interact with activated ELK, and that activation of ELK will lead to transcription of CFOS. CFOS is given rise to because of this interaction of LAT with GRAB2 SOS. This is one part of our AP1. We need two parts, CFOS and CJUN. So CFOS was from RAS interacting with the GRAP2 SOS to give rise to RAS GTP. And this RAS GTP will eventually lead to the activation of ERK along with activated ERK, both together will lead to transcription of CFOS. At the same time, this LCK binding to the phosphorylating the zeta chain leading to the interaction with ZAP70 and LAT will also give rise to similar interaction where there is another adapter molecule instead of GRAP2 SOS called VAV. And that VAV, instead of interacting with RAS GDP, will interact with RAC GDP, and that will give rise to RAC GTP. RAC GTP will interact with JUNK, and then that JUNK C-Jun N-terminal kinase or SAP kinase will be activated, and the activated JUNK will phosphorylate a molecule called C-Jun. C-Jun and C-Phos together are going to give rise to activated AP1 transcription factor. This transcription factor will then go to the nucleus and help in promoting transcription of certain genes that are important for T cell pathway. Questions? All right. So this was one pathway, MAP kinase pathway, that happened because of after LAT, LCK ZAP70 LAT, the two important molecules are GRAP2 SOS leading to formation of RAS GTP and VAV leading to formation of RAC GTP. RAS GTP leads to formation of CFOS and RAC GTP leads to formation of C-Jun. C-Jun and CFOS come together to give rise to AP1. All right. Now we're going to talk about another pathway. This is PLC gamma pathway, and this diverges into two different pathways, IP3 and DAG. So again, the initial part is the same, LCK, ZAP70, LAT. Now that LAT can interact with something called ITK. In B cells, that's BTK. You have heard of BTK. We don't talk about ITK as much, but here is our ITK in T cells. Oh, sorry, if you can see this molecule here. So ITK, when that LAT interacts with ITK, ITK can interact with PLC gamma 1. PLC gamma 1 is in T cells, PLC gamma 2 is in B cells, I believe. We'll come back to that. So PLC gamma 1 is going to give rise to that in PLC gamma 1 will act on this PIP2 to give rise to DAG and IP3. DAG is going to go into PKC, is going to interact with PKC protein kinase C, and that's your NF-kappa B pathway. We'll come back to that. We are going to now talk about IP3 pathway. So LAT interacts with ITK. ITK helps, it's a kinase, it phosphorylates PLC gamma 1. Phosphorylated PLC gamma 1 is now going to act on PIP2 and phosphorylate it so that it gives rise to DAG and IP3, inositol triphosphate, right? This IP3 is going to be important because it leads to, let me see quickly, it leads to this whole pathway that is important for calcium molecule, and that interaction will lead to NFAT pathway, NFAT transcription factor activation. How does this happen? So this IP3, once activated, it's going to interact with endoplasmic reticulum, and that endoplasmic reticulum is going to release calcium from the endoplasmic reticulum into the cytoplasm. When that calcium is released into the cytoplasm, the endoplasmic reticulum has less calcium in it. There is a sensor called STIM1, this pink thing here. It senses that the calcium is low in endoplasmic reticulum and goes ahead and interacts with the crack channel that's present in the cytoplasmic membrane of T cell. When that crack channel, which has a protein called ORI, opens up, there is extracellular calcium that's going to be coming into the T cell. That calcium, increase in calcium, is going to lead to various more interactions, and let's talk about that. All right. So once that calcium comes in, it interacts with calmodulin, and calcium calmodulin dependent calcineurin phosphatase is going to be activated, so that it activates NFAT. NFAT is a, all right, moving back here, sorry. Here's the calcium. So calcium makes a bond with calmodulin and interacts with something called calcineurin. And that calcineurin phosphatase will activate NFAT. NFAT is going to move to this nucleus and help in interaction with the promoter region of various genes such as IL-2 that will lead to T cell function. So this interaction between calcium calmodulin and calcineurin, it's important because calcineurin is a phosphatase that will dephosphorylate NFAT, and dephosphorylation of NFAT actually activates it in this case. Important to remember because there are some immunosuppressants that use this pathway to suppress the immune system. So calcineurin inhibitors such as cyclosporin will interact with this so that that phosphatase is not active, so that T cell is not activated from NFAT pathway. So cyclosporin and tacrolimus are two drugs that are important and use this pathway for immunosuppression. All right, and then the third pathway is our DAG pathway. DAG, moving back here, see, this is the pathway LCK ZAP70 LAT. LAT interacts with ITK. ITK phosphorylates PLC gamma 1. PLC gamma 1 phosphorylates PIP2 and interacts with PIP2 to give rise to DAG and IP3. DAG activates PKC. This PKC, sorry, where is that? Okay, PKC will interact with I-kappa B kinase, and that will lead to activation of NF-kappa B, and that NF-kappa B will move to the nucleus and help in transcription of certain genes, right? NF-kappa B is not only active in T cell signaling, it's also important for TLR signaling, innate pathways, cytokine signaling. So NF-kappa B is a major signaling pathway that is downstream of several other immune molecules. So just kind of keeping that in mind. So regardless, now we know there are three pathways for T cell receptor signaling that are important. MAP kinase pathway that leads to formation of CFOS and CJUN that gives rise to AP1. There is another pathway that is calcium dependent, which is NFAT, and then NF-kappa B after PKC activation. So that was our T cell receptor pathway signaling. Important to remember is the regulation of T cell signaling. We need to make sure that once it's activated, there is a way to curb T cell signaling so that there is no inflammation and ongoing damage once the T cells have done their job. The regulation occurs at several levels. So at the level of genes, there is regulation of T cells using non-coding RNA, such as long non-coding RNA and microRNA. These non-coding RNA do not make proteins, but when they are created, these RNAs are important in regulating the mRNA activity. So microRNA particularly are formed as a primary transcript, and then they are cut up from the primary transcript. There are some enzymes or proteins called TROSHA and DICER that make sure that they cut up that pre-mRNA to microRNA or miRNA that interacts with mRNA along with these proteins called argonucleotide proteins. And they form a complex called RNA-induced silencing complex, or RISC, and it silences the mRNA. So it will say, for example, if it silences the IL-2 gene, then the T cell is not going to get the positive feedback from the IL-2 gene or IL-2 protein, so that it will not lead to increased survival and it will not lead to activation or differentiation and things like that. So it helps in regulation of T cell signaling. That was at the RNA level. Now we talk about if the T cell is present, there is IL-2, it's doing its job, and the receptor signaling pathway is activating the T cells. There are phosphatases. Remember, the T cell signaling has all these tyrosine kinases that help in phosphorylating the items and leading to activation. There are several phosphatases that help in regulation called SHP1, SHP2. So these dephosphorylate are the regulating part of the tyrosine kinases, whereas SHIP helps in curbing the action of PI3 kinase. There is also a phosphatase called CD45, such as LCK and FIN, that have important role in actually starting the activation of T cells. We'll come back to that. There are other molecules called CD2 family, CD2 SLAM co-stimulator. CD2 binds to LFA3, and then there is a protein called SLAM. Instead of having ITAM or ITIM, ITAM has activating motif, ITIM has inhibitory motif, and the ITSM is called switch motif, which means initially it can be an activating and then later it can turn into an inhibitory motif. So ITSM is attached to this protein called SLAM, and there are two kinds of SLAM, SAP and XLP. These proteins, SAP and SH2-DIA, these are proteins, if they are defective, they lead to X-linked lymphoproliferative syndrome, type 1 and type 2. So there are several other proteins, molecules in this family, CD2, SLAM, 2B4. 2B4 also has that switch motif. This is not from Abbas, but I just want to make sure that you understand what happens with LCK. So remember, CD4 and CD8 at the cytoplasmic tail has LCK. LCK is in an inactive stage, but when the CD4 is engaged, when it interacts with the ligand and MHC in the antigen presenting cell, this structure that is an inactive form of LCK will be activated. What happens is there is a tyrosine that is phosphorylated, and the phosphorylated tyrosine in case of this molecule inactivates it. So it keeps it inactivated. When there is LCK that is activated, when its terminal tyrosine is dephosphorylated. So when it gets dephosphorylated, it untwines from this whole bound structure so that it's available to bind to the tyrosine on the zeta chain. So just FYI, CD45 is important, where actually the dephosphorylation or phosphatase takes part in activation, whereas the phosphorylated form is the inactivated form. So the kinase will lead to inactivation, whereas the dephosphorylation will help in activation. All right, there are some metabolic changes during T cell activation that occur where in a resting stage of T cells, typically it uses oxidative phosphorylation. But when the T cells are proliferating, it may use aerobic glycolysis for its metabolism. Oh, this is actually another slide that actually shows most of the pathways that we just talked about. All right, I'm going to stop here and ask for questions. I'm wondering, Ishan, was it you that you had sent me this slide last time and I added it to our discussion because it has a lot of information that we talk about?
Yeah, I think so, Dr.
Yeah. So thanks for sending that slide. It's really important because this is a really nice slide because it kind of puts everything into perspective of what happens in terms this is all happening simultaneously. Here's our TCR, zeta chain ZAP70, and then here is your IP3 and DAG, PI3 kinase, that's helping in keeping the cell alive. And then the DAG will lead to NF-kappa B pathway, your ZAP70 working with GRAB2SOS, leading to your FOS, and somewhere there should be, here is your June, CFOS and CJune, so your MAP kinase pathways helping in AP1, and then your calcium pathway leading to your NFAT. So all of these pathways coming together. All right. Questions. We talked about T cell signaling. That's our major pathway. Now we are going to use B cell signaling to just kind of point out the differences. So if you have questions, I think this is a good time to ask those. All right. Let's talk about B cell signaling. Pretty similar concept. There are few differences. In case of B cells, the membrane immunoglobulin is your BCR, and it has two signaling molecules, Ig alpha and beta. Each one have one item on their tail. This B cell co-receptor, B cell receptors, co-receptor is CR2, CD19, CD81. These are interacting with the same antigen that is being that is going to attach to the BCR. So say a complement protein has tagged an antigen, and it has its complement particle or C3d attached to it, and here's the antigen. That antigen is, because it's tagged, now it's going to interact with the B cell receptor. But along with that, the complement particle will interact with the co-receptor using the CR2. CR2, downstream of that co-receptor are our Circ family kinases, such as LIN. LIN will phosphorylate SIC, which is gonna, here is our LIN. Okay, so LIN, FIN, or BLK will interact and phosphorylate the signaling chains. And once that happens, there is gonna be another adapter protein called SIC, or a kinase called SIC instead of ZAP70, that will interact and help in activation. So the next step is LIN, SIC, here is, all right. So moving on, LIN, SIC, and then that interaction goes in further for PI3 kinase that will help in keeping the B cell alive. All right, so going back to this interaction, when the B cell binds to an antigen, it will lead to cross-linking of several BCRs on the surface of B cell. The co-receptor BCR, as we saw, will phosphorylate the items on the co-receptor. So the co-receptor here has LIN, FIN, and bulk BLK, which will phosphorylate the items on the signaling proteins. Once that happens, SIC comes along, and SIC helps in phosphorylating the next step, which is your SLP65 or BLINK, B cell linker protein. It's two names for the same thing, SLP65. So this is just like LAT that we talked about. So instead of LAT that was in the bound to the membrane, this is more cytoplasmic, where once it interacts with SLP65, there are multiple different molecules. It's an adapter protein, so multiple different pathways get activated. SLP65 can interact with GRAB2SOS that will help in RAS and RAC. GRAB2SOS and VAV, they help in activation of RAS and RAC GDP to RAC GTP and RAS GTP. That will lead to ERK and JUNK and lead to CFOS and CJune formation and activation of AP1 transcription factor. Also, there is this BTK bound to SLP65, and BTK will phosphorylate PLC gamma. PLC gamma activation will lead to DAG and IP3, DAG will lead to activation of protein kinase C that will help in NF-kappa B activation. That is a transcription factor that will go to the nucleus and help in transcription of certain genes that are important for B cell function. And then PLC gamma activation will also lead to IP3. IP3 will lead to increased cytoplasmic calcium. The calcium is low in the endoplasmic reticulum, so will activate STIM, will open up the crack channel, will increase the calcium in the cytoplasm, and that leads to calcium calmodulin interaction and activation of calcineurin bound to the that will activate the NFAT, and that interaction will lead to activated NFAT going to the nucleus to help with transcription. Another pathway that's mentioned here is MYC. Not going to go into the details of that, just pointing out certain important molecules here. We talked about LIC, ZAP70, and LAT for T cells. Similarly, there is LIN, SIC, and BLINK in case of B cells. LIN, SIC, BLINK, right? And then instead of ITK, there is BTK in B cells. Instead of PLC gamma 1, there is PLC gamma 2 in this case. So PKC theta in T cells, PKC beta in case of B cells. So very similar signaling, just different molecules end up in activation of different same transcription factors that go to the promoter region and help in transcription of genes that are important for B cell function, not the T cell function. So there are few differences. All right. We talked about the signaling. Now we talk about some inhibitory receptors that are important for attenuation of immune receptor signaling that we just talked about. So those are CTLA4, which is an important inhibitory receptor that can bind to the co-stimulating molecules B7 proteins B7-1 and B7-2, but it will help in inhibition. Similarly, PD-1 can bind to PD-1 ligand, and that has inhibitory ITIM kind of receptor, so will lead to inhibition rather than activation. Some other inhibitory receptors include CD94, NKG2A, HLA-E in case of NK cells, several KERs that are in NK cells, Fc gamma R2b for B cells, CD22 Siglec2, which are important for B cell signaling attenuation. We talked about all this phosphorylation. There are also another way of attenuating this whole pathway by something called ubiquitin ligases. What does it do? Ubiquitination, which means tagging for destruction. That's what ubiquitination does. So what it does is when there are activated molecules, say for example, the signal receptor has already bound to the ligand, this LCK has already activated or phosphorylated the items on zeta chains, and ZAP70 has been engaged. There is a way to recruit ubiquitin ligase such as CBLB to activated ZAP70, and that ZAP70 will be tagged for ubiquitination, which means it's going to break down some of these parts so that they are going to undergo degradation in lysosomes, in the lysosome. So that's your ubiquitination that can be used. All right, again, stopping here, go ahead. All right, now we're going to talk about cytokine receptors and their signaling. So we know that there are several cytokine receptor families. These are based on the structure of these cytokine receptors and some of the pathways that they use. So the type 1 cytokine receptor families all use JAK-STAT pathway for their signaling, and there are 3 subgroups. These are common gamma chain. All right, let's see where that is. Common gamma chain, common beta chain, and GP130. These are the type 1 cytokine receptors. All these cytokine receptors have certain things in common. They have this structure that is, or conserved cysteines that are common to all of these 3 subgroups. But then The common gamma chain, as the name suggests, has the common gamma chain in each of them. The common beta chain has the beta chain, which is part of their structure, and then the GP130 chain is present in the third group. So important to remember is common gamma chain is present in 2, 4, 7, 9, 15, 21. There's no connection here. Somehow you guys have to memorize these numbers. It's really important to remember these cytokines 2, 4, 7, 9, 15, 21. The important clinical tidbit to that is that common gamma chain defect leads to severe combined immunodeficiency, which is X-linked, and one of the most common causes of skin. So that's your common gamma chain. The common beta chain is present in IL-5 and GM-CSF. GP130 is present in IL-6, IL-11, IL-27. All right, apart from that, now we're going to go on to talk about type 2. They also use JAK-STAT pathway. These include interferon alpha and beta, interferon gamma, IL-10, IL-20, IL-22, IL-24, 26. All of these are type 2 cytokine receptors. They also use JAK-STAT pathway for signaling. Then there is this whole family of TNF receptors, including TNF alpha, TNF beta, CD40, Fas ligand, BAF, and there are all these mentioned here. All of these are part of TNF receptor family. IL-1 family includes IL-1 and IL-18, and these are important for inflammation, inflammasome formation, so kind of important to remember in terms of autoinflammatory syndromes. IL-17, 17A, 17B, C, D, E, IL-17F, all of these are part of IL-17 receptor family, important for very early onset IBD kind of patients. 7 transmembrane GPCRs, these are important receptors in terms of chemokines, histamine, all of those. So again, all these receptor families have their own way of, they have common structure to each of these families, and then they have their own way of receptor signaling. Let's talk about TNF receptor signaling. TNF receptor family, as I talked about, has multiple different types of receptors. TNF receptor type 1, type 2, lymphotoxin, CD40, Fas, BAF, all of these receptors. These are trimers. So we talked about TCR or PCR, where there are 2 chains that come together to form the TCR or BCR, whereas these are trimers. There are 3 molecules or 3 chains that come together. And also they bind to the ligands that are also trimers. So TNF receptors bind to TNF, which are also trimers. The ligand itself is a trimer. And then similar to what we talked about in our TCR and BCR signaling, there is that outer extracellular portion that binds to the ligand. That's the first phase. Then there is that second phase, which is the cytoplasmic phase. Here, there are multiple adapter proteins. There are multiple enzymes that take part in activation of transcription factors. And those transcription factors then go to the nuclear phase and help in transcription of certain genes important for TNF receptor function. Right? So here, once the trimer of TNF binds to the trimer TNF receptor, there are multiple downstream adapter proteins and enzymes. One of them is called TRAD, T-R-A-D-D, TRAD, and TRAD leads to the interaction of something called TRAF. TRAD binds to TRAF, and TRAF leads to multiple pathways. MAP kinase pathway leading to AP1, I-kappa B pathway leading to NF-kappa B activation, and caspase 8 pathway. What does TNF receptor typically do? It leads to apoptosis. So these are also called death domains. These TRAD domains are also called death domains, and that caspase 8 activation leads to activation of effector caspases that leads to apoptosis. Another pathway is JAK-STAT JAK-STAT pathway. JAK-STAT pathway has the JAK and the STAT. JAK are different types. JAK1, there are 4 JAKs, JAK1 to 3 and TYK2. Don't ask me why they named TYK2 instead of saying JAK4, but TYK2 is another type of JAK. Then there are STATs. There are 1 to 4 STATs, 5A and 5B and 6. So those are your 7 important STATs. What does STAT stand for? Signal transducers and activators of transcription. So that's your STAT, and JAK stands for Janus kinase. So it is a kinase, which means you would automatically know it adds a phosphate molecule to whatever moiety it interacts with. So let's see what happens with that. So here is your cytokine receptor, 2 different chains. When the cytokine comes along, that ligand helps these cytokine receptor chains to come close to each other, and then the cytokine binds to it. When that happens, in the cytoplasmic tail are our kinases. So the JAK are bound to the cytoplasmic tail of the cytokine receptor. That helps in phosphorylating. Once that ligand is bound, it helps in phosphorylating the receptor chain. Once that phosphate is added, it leads to this inactive STAT to interact with that phosphorylated receptor chain. So the STAT is recruited to the receptor cytoplasmic tail of the receptor. When 2 STATs come together, come in close proximity, once they are phosphorylated, the JAK also phosphorylates the STATs, and when they phosphorylate the STAT, they dimerize, which means the 2 molecules come close to each other to form the active STAT. That STAT is your transcription factor. It goes to the nucleus, and it helps in, once it's translocated to the nucleus, it binds to the promoter region of specific genes so that it can lead to the transcription of those genes that help in the function of that cytokine, right? So remember, we always connect back the ligand binding to the end result. So depending on the cytokine that interacted with its receptor, JAK-STAT pathway will lead to activated STAT going to the nucleus, binding to the promoter region of the genes that are important for the function of that particular cytokine. So that's your JAK-STAT pathway when it's activated, but it of course needs negative regulation like any part of the other immune system that needs regulation. So there are suppressors of cytokine signaling, which means SOCs, S-O-C-S, so suppressors of cytokine signaling. They help in making sure that the JAK-STAT pathway, once it's activated, is able to be suppressed. SHP1 and SHP2 are important for inhibition of that tyrosine kinase activity. So those are also they will also help in regulation of JAK-STAT signaling. And then the third one is protein inhibitors of STAT, or PIAS. Protein inhibitors of STAT help in deactivating this STAT pathway. All right, one more pathway, a really important pathway, is your NF-kappa B pathway. There are 2 types of pathways: canonical pathway, which is your typical pathway that we talk about for NF-kappa B signaling, and the non-canonical pathway. Now, if you look at this, the NF-kappa B itself has parts of its structure called REL-A and P50. That's your canonical pathway and NF-kappa B. Along with that, in the non-canonical pathway, there is another type of NF-kappa B which has P100 molecule that binds to REL-B. That's your non-canonical pathway, NF-kappa B. Keep that in mind. That's just the NF-kappa B. Now we're going to talk about signaling. In general, I think of NF-kappa B as a captive. It's captive. It is captured by its inhibitor. The inhibitor of kappa B is called I-kappa B alpha. All right. So our NF-kappa B is captive because of its inhibitor. Because it's bound to its inhibitor, it's in its inactive state. Sorry, give me 1 second. I need to take 1 minute. I'll be back. Sorry, guys. Rizal wanted to bark. All right. So, where were we? So, our NF kappa B is captive because of its inhibitor. So, it's in its inhibited state and it needs to be released from its captive. So, from its captive state. So, there is an enzyme called IKK. It's a kinase that has to release this from its inhibitor. All right, with that background, let's talk about this pathway. So this pathway is downstream of multiple receptors such as TNF receptor, TLR pathway, TCR pathway, BCR pathway, multiple pathways. So what happens is, depending on the type of pathways, there are different molecules that interact with each other to activate this enzyme complex called I-kappa B kinase, which IKK stands for I-kappa B kinase, and it's a complex of three parts: IKK alpha, IKK beta, and IKK gamma. IKK gamma also is known as NEMO, right? And this whole complex comes in contact with that inactivated NF kappa B so that it can release the inhibitor, and so that once the inhibitor is remember this is a kinase, so it adds a phosphate to the inhibitor of kappa B and ubiquinates it. So the IKK gamma or the NEMO is the part that's the active part of the kinase, and alpha and beta are needed for just the regulation of this kinase. Once that inhibitor is phosphorylated, it is ubiquinated so that it goes into the proteasome and gets cut up into pieces. At the same time, when that is released, the inhibitor is released, the NF kappa B is activated. That activated kappa B will go to the nucleus, bind to the promoter region of particular genes to help with the transcription of certain genes important for that particular receptor that helped in activation. All right, so this was the canonical pathway. There is also a non-canonical pathway. Instead of IKK alpha, beta, and gamma molecules, there are two molecules of IKK alpha that come together to activate the NF kappa, non-canonical NF kappa B. And once ubiquinated, the inhibitor is ubiquinated, the kappa B is released, and the activated form of kappa B goes to the nucleus. Really important pathway, and to memorize, I think again the other important part of that is the NEMO part of this kinase if there is a defect in that gamma chain of the IKK, it can lead to NEMO defect that can present with severe life-threatening infections and other features. TGF-beta pathway signaling. So TGF-beta is another cytokine. If this here is its receptor R1 and R2, and they can interact. So here is the TGF-beta that will bind to its receptor. When it's bound to its receptor, there is something called receptor SMAD that comes and attaches to the receptor cytoplasmic tail and will get phosphorylated. Once phosphorylated, the co-SMAD can bind to that R-SMAD. R-SMAD and co-SMAD form a complex that is a transcription factor, will go to the nucleus and help in transcription of the target gene. Questions? Right. That was the end of our chapter. Going back to the questions, which of the following is an example of GPCR?
Is this CCR7E?
CCR7 is correct. And in the T cells calcium signaling pathway induced by antigen recognition, which enzyme and transcription factor are activated?
It's calcineurin.
This E.
Yep, calcium.